Hey guys, welcome back to One Stop Biology. So guys, in today's video, we are going to understand plant growth regulators, right? So we are right now at chapter 15 of class 11th biology, and we have already understood the meaning of growth. We have understood the meaning of development, right? And we are going to understand what are the factors. So in the last video, we understood there are some factors that actually affect the growth of the plants. These factors can be intrinsic factor and extrinsic factor. Now, before we start off, guys, if you're new to the channel and you have not subscribed to the channel yet, please do subscribe to the channel and press the bell icon to get all the updates. And yes, if you have any doubts, if you have any feedback, do keep giving your feedback in the comment section of the video. So guys, when we talk about the growth or development factors, in the last video, the first thing we understood is that these are of two types, intrinsic and extrinsic. And then we also understood that these intrinsic factors are basically divided again into two, intracellular and intercellular. Now, intracellular are actually genetic factors, and we are not going to talk about this. Rather, we are going to talk about the intercellular factors like plant growth regulators. So, the topic plant growth regulators is actually part of intercellular intrinsic growth, right? We are going to understand about this. Now, when we talk about the characteristics of plant growth regulators, guys, actually plant growth regulators or PGRs, they're also known as PGRs. So basically PGRs, these are basically small simple molecules and it can be of diverse chemical composition. So they are small simple molecules of diverse chemical composition, right? And they could be basically different type of derivatives, chemical derivatives. So say they could be indole derivatives, like we have IAA, indole acetic acid, right? They can be adenine derivatives, right? Like we have kinetin, right? They can be carotenoid derivatives, derivatives like abscisic acid, right? They can be terpenes, right? Gibberellic acid. They can be gases as well. They can be gases as well, like ethylene, right? So basically, these are chemical compounds. These are small and simple molecules, and they can be derivative of different kind of chemicals. They can be a derivative of indole, adenine, carotenoids, terpenes, or gases as well. Now, they are basically described as plant growth substances or plant hormones. They are also known as plant hormones or phytohormones, right? So these are also known as plant growth substances, phytohormones, or plant hormones. Now, if we see the division of PGRs, if we see the division of plant growth regulators, you'll see that they are divided into different types. So basically, PGR, plant growth regulators, are divided into two groups based on the function. What is their function in the living plant body? So the first group of PGR are basically involved in growth promoting activities. They promote growth. Okay, such as cell division. Cell division is a kind of growth, right? Cell enlargement, trophic growth, fruiting, flowering, or seed formation. Seed formation, right? Now, these are also known as plant growth promoters. These are known as plant growth promoters, right? So, the first group of PGRs are the growth promoting group, right? So these are known as plant growth promoters, and these take part in several growth activities, like cell division, cell enlargement, pattern formation, flowering, fruiting, seed formation, right? The example of this is auxin, gibberellins, cytokinins, right? Now the other group, PGR of the other group basically plays an important role in responses, plant responses. Now this can be towards wounds, towards stress, right? Such as say dormancy, seed dormancy, or abscission, right? So basically example is abscisic acid. AB, right? Abscisic acid. That belongs to the group where they respond to different type of stress, wounds. Now, the gaseous PGR, like ethylene, right? Again, fit better to the second group, right? Or say the gases one can also fit to the first group as well, plant growth promoters. So that is why we say that the gaseous PGR like ethylene could fit either of the groups. It could fit both the groups, and it is largely an inhibitor. Basically, it is an inhibitor of growth activities. So there are certain cases where we need to inhibit these activities. We need to inhibit the activities of promoters, growth promoters. And there we basically use inhibitors of these activities, right? So again, whether it's promoters or whether it is inhibitors, both are part of plant growth regulators. That is the first thing. Now let's see how it was discovered, how exactly these plant growth promoters were discovered. Now guys, plant growth promoters or plant growth inhibitors, or in general I can say PGRs, plant growth regulators. Discovery of each of the five major groups of PGR have all been accidental. So basically, it started off with the observation of Charles Darwin, right? You all must have heard the name of Charles Darwin and his son Francis Darwin. So they were basically observing that the coleoptiles of canary grass responded to some illumination, unilateral illumination, by growing towards the light source. So it used to bend towards light basically, right, and grow towards it, right? So basically it was concluded that the tip of coleoptile was site of transmittable influence that caused bending of the entire coleoptile, right? Now what happened was later auxin was isolated by F.W. Wendt. So F.W. Wendt isolated auxin from the tip of coleoptiles of oat seedlings, right? And after that, what happened was again a different scientist, right? Kurosawa, right? Kurosawa. What he did was he basically saw the disease of rice, the disease in rice seedling, the disease is known as bakane, foolish seedling, was caused by a fungal pathogen, Gibberella, Gibberella fujikuri, right? So Kurosawa reported the appearance of symptoms of a disease in rice seedling when they were treated with sterile filtrates of the fungus itself, and the active substance was gibberellic acid. Again, in the similar way, F. Skoog and his team observed from the internodal segment of tobacco stem. So from tobacco stem, which is the callus basically, callus is what? It is undifferentiated cell, a mass of undifferentiated cell. So that proliferated only if we added oxygen in the nutrient medium, right? So when oxygen was supplemented, they used to grow, right? Now, what happened is that it was one of the following extracts of vascular tissues. It was also in yeast extract, it was also in coconut milk or DNA, right? And later on, Miller in 1955, remember, Miller in 1955 later identified and isolated the crystallized form of cytokinins, right, which promoted active substances which they termed as kinetin, right? Now after that, during mid 1960s, three independent researchers, they reported the purification and chemical characterization of three different kinds of inhibitors. The first was inhibitor B, the second was abscission 2, and the third was Dormin. Later, all three were proven to be chemically identical and they were named abscisic acid ABA, right? In the similar way, H Cousins in 1910, right, in 1910, confirmed the release of a volatile substance from ripened oranges, and that ripening of stored unripe bananas as well. Later, this volatile substance was identified as ethylene, a gaseous PGR, and after that, after discovery of all of these different type of chemical compounds, people started studying their effect as well, and all the different type of effects of these compounds we are going to cover in the next video. So we in the next video we are basically going to cover the physiological effect of plant growth regulators. I hope you would have understood the basis of plant growth regulators, how they are divided, what are different kind of groups involved, chemical groups involved. So guys, please do like the video and share it with your friends. And if you're new to the channel, please do subscribe to the channel and press the bell icon to get all the updates, all the notifications. And yes, if you have any doubt, please do not hesitate in asking your doubt in the comment section of the video. Thanks for watching this video, guys. See you in the next one. Till then, take care. Bye-bye.